So today's lecture is about nutrient uptake and transport. Basically, we already discussed about the basics of nutrient acquisition and then also the basic concept of nutrient uptake. Today we'll talk further about the uptake uh, from the epidermal cell, the plasma membrane, and then the short distance transport, mean the transport from the epidermal cell to xylem vessels. So that is basically a horizontal movement and is also called as the short distance transport, which is from epidermal to xylem vessels. The first of all, we need to know that there are some kind of transport proteins which are present, which we have already discussed in the previous lecture, that these proteins are basically the facilitator of the ions to get into the symplast. So these are also the barriers because these are specific and every ion or all the ions cannot pass through these proteins. But many of the ions they can pass through either because they are either specific to one ion or they may be specific to more than one ion. But it doesn't mean that when we say more than one ion, that all the ions can pass through it because these are specific to specific ions. Maybe two, three, four ions may pass due to their commonalities, but all the ions will not pass through. So we will talk about these proteins in this lecture. So ion transport through plant cell membrane proteins. Transport across plant membrane is facilitated by transmembrane proteins. These can be classified into three groups. The first, uh, the primary active transport, which is through pump. Then this is directly coupled with the use of energy in the form of ATP. And then the second, that is secondary active transport or coupled transporter, which harness the electrochemical gradient of generally H ions to the movement of solute uh, in either the same or in the opposite form. So it means directly these pumps they doesn't use energy or these transporter they doesn't use they do not use energy in the form of ATP, but they utilize the electrochemical gradient which is developed by the use of ATP. So it means this is also a kind of active transport. But because the directly energy is not used, so that's why we say secondary active transport. So there are also passive transporters which catalyze the movement of solute down their electrochemical gradients from higher concentration to lower concentration. In these cases, again, there may be the natural gradient that is mostly in the start of the uptake process, but later on, while the inside the cell, the concentration is increased, so the movement or the passive movement is not possible. So in that case, then again, the electric gradient is developed by these pumps, and then that is utilized by these uh, ions. The later group includes a variety of carriers and channels can be distinguished from uniport carriers by the high catalytic rate, which can exceed 10 million ions per second, which is several orders of magnitude greater than uniport. Uh, Here you can see the different kind of proteins which may be in the cell membrane, and there are mentioned. symport, and antiport, and uniport. So I think we should stop because there is Azan. So this figure then can explain very briefly that what is primary active transport, what is secondary active transport, and what is passive transport. And these are related with the with the proteins or with the transporters. Then the primary active transport is mostly that is in pumps because these pumps they utilize energy in the form of ATP and they pump out proton. Mostly it is important for the plants, but also calcium and other ions. When we say secondary active transport, then in that, because I mentioned that a gradient is developed and that gradient is used to transport the ions. For example, the symport, there is a symport of potassium and proton. It means when proton gradient is developed, this proton is used for symport of potassium. This proton will come along with potassium and will transfer potassium from one side to the other side. And this proton on the other side of the cell membrane is uh, released by use of ATP through proton pumps. So it means the primary active transport of proton, uh, which release proton to the other side and develop gradient. That gradient was utilized for the movement of potassium from that side to the inside and in the form of symport. But there can also be an antipode. For example, uh, if the proton gradient is developed outside the cell membrane, they can transport with the sodium in the form of antipode because uh, plant need to spell out the sodium from uh, inside to the outside. क्योंकि sodium जो है that is toxic for the plant cell metabolism. So in that case, there this is kind of antipode because the protons they are coming in and sodium is coming out. Then we have a unipode. In unipode, they allow the movement of ion from one side to other side by using that gradient or by using the natural gradient or natural chemical concentration gradient of that ion. So then these are basically the transporters. We can say these are carriers. So we'll see in the next perhaps that what is difference between carrier and pumps and channels. So it means the carriers, they can perform three kind of transport: symport, antiport, and uniport. But again, we have channels, and the channels mean we have a transporter which can uh, transport millions of ions per second. So these are kind of open, you can say like hole type structure, and the bulk of the ions they can pass through. But even then, when we say these are like open hole, it doesn't mean they allow everything to get into the cell because uh, these are also specific. These will allow only for which they have a mechanism inside. Uh, the one thing I missed here that the passive transport is either through channels or through carriers. It means the carriers are the transporters which can have secondary active transport or they can be utilized as a passive transport. So both type of transport of ions done through uh, can be done by these carriers. So this is another example here mentioning the previous, which we have discussed about the electrochemical gradient. Basically, the driving force of any ion, so that is either the solutes, mean chemical concentration. Chemical concentration, if we say that पोटेशियम की है तो पोटेशियम आयन कंसंट्रेशन, अगर कैल्शियम है तो कैल्शियम, अमोनियम है तो अमोनियम. So it means if one side has a higher concentration of ammonium ion and other has lower concentration, they can move easily. Uh, and the other thing that is the charge. These are positive charge. For example, if the positive charge at one side is higher and on the other side of the membrane is lower, so the ions mean the nutrient, they can pass or they can move from one side to other side by using that charge gradient. क्योंकि अगर हम पोटेशियम को देखें या कैल्शियम को देखें या अमोनियम को देखें तो उनके ऊपर भी पॉजिटिव चार्ज होता है. तो अगर दूसरी साइड के ऊपर पॉजिटिव चार्ज कम है तो एक साइड के ऊपर पॉजिटिव चार्ज ज्यादा है तो वो न्यूट्रिएंट्स जो है दे कैन मूव फ्रॉम दैट मोर पॉजिटिव चार्ज टूवर्ड द लेस पॉजिटिव चार्ज. अब वो ज्यादा पॉजिटिव चार्ज क्यों डेवलप हुआ है, तो वो प्रोटॉन की एक्टिव जो ट्रांसपोर्ट है, जिसको हम प्राइमरी एक्टिव ट्रांसपोर्ट हमने पिछली स्लाइड में कहा है, तो उसकी वजह से हो सकता है. So it means that gradient or that charge gradient is used for the movement of ion. Now, coming toward these uh, channels and the carriers. So the simple diffusion is like that the ions they pass through the cell membrane, the lipid bilayer. But we know that the ions they cannot pass through the lipid bilayer. But the molecules they can pass through the lipid bilayer. So this is a kind of simple diffusion. When we say the proteins are there, so we say these are facilitated diffusion. And when we say channel, it means there is an open hole, a bulk of ions they can pass through. But when we say carrier, so carrier means the ion from one side will attach to the carrier and then it will be moved to the other side by changing conformation. It means the process is slow because the ions they need to attach on one side of the protein and then that will be released on the other side. So it means it will take time. So the movement through carrier is relatively much slower as compared to channels. So under most circumstances, the driving force for the facilitated diffusion of an uncharged solute across a membrane is its concentration gradient, as I mentioned earlier. Whereas for an ion, it is its electrochemical gradient. जब हम ion की बात करेंगे, तो then the electric gradient will also be along with. If something is solute, like they are not ions, they are compounds, they are molecules. So then the charge gradient will not help. But when there are ions and the electric gradient can also help for the movement of those nutrients from one side to the other side. So passive uptake along with the electrochemical gradient, then active uptake against electrochemical gradient. So I think we have already discussed about it, and all these has been explained. So now coming toward the channels, ion transporters channels, these are transmembrane proteins that function as selective pores. They are pores but selective, form an open water-filled channel across the membrane. Transport through channel is always passive. And transport through channel is fast, that is 10 raised to power 8 molecule per second. And another thing is that there is a steep increase in the flow. So when you enhance the concentration of those solutes in the solution, then the of course the movement will be linearly enhanced because they are not because there is no kind of what you say the attachment of the molecule with the protein. And as I mentioned that these are the selective pores, it means the higher the concentration of that ion, they can move very easily through these pores. So I mentioned that these are specific pores. They are not open to every ion. So the opening and the closing of these pores depend on various factors. For example, there are channels which depend on the voltage. The difference in the voltage on the both side of the membrane, so that determines that whether the channel will open or not. Sometimes there are specific legends. These legends can be from the outside and these can also from the inside. These are specific, but can you can say these are kind of specific stimulators which will fix to the proteins or channels, and then these channels will open. Unless these are not attached to these proteins, these will not open. So sometimes there are kind of a mechanically gated, they require some kind of a specific pressure, and that pressure is developed by ions because most all the ions they have their specific or their atomic weight. So based on that, so they develop some kind of mechanical pressure, and that mechanical pressure will, of course, open that channel. So this is very clear that these specific pores, they are specific and they are not open to everything. Then we have also the voltage gated ion channels are class of the transmembrane proteins that form ion channels that are activated by change in the electric membrane potential near the channel. And the membrane potential alters the conformation of the channel protein, regulating their opening and closing. As I mentioned earlier, that the basically the electrical membrane potential, so that varies or that determines, and that is due to the charge. क्योंकि जो मेम्ब्रेन के इर्द गिर्द जो है वो चार्ज या आयंस जब आते हैं तो वो उसका जो इलेक्ट्रिक पोटेंशियल है उसको वेरी चेंज करते हैं. तो जो स्पेसिफिक होती हैं उस मेम्ब्रेन पोटेंशियल के लिए वो ओपन हो जाती है. तो इस तरह जो इलेक्ट्रिकल मेम्ब्रेन पोटेंशियल है वो उनको ओपन करने में हेल्प करता है. So there are, as mentioned, there are legend gated ion channels, uh, also commonly referred to as ionotropic receptors, are a group of transmembrane ion channel protein which open to allow ions such as sodium, potassium, calcium, and chloride to pass through the membrane in response to binding of the chemical messenger. जो मैंने आपको स्टिमुलेटर कहा है दैट काइंड ऑफ द केमिकल मैसेंजर है, जब तक वो अटैच नहीं होगा ये ओपन नहीं. तो वो जो मैसेंजर है तो दैट इज डिमांड बेस्ड. Sometimes the kind of receptor. So they basically develop a messenger, and these are messengers are basically chemicals. We say ligands. So they attach to that those ions, and then they allow those specific ions to pass in. There are, I mentioned that there are some mechanical base, and this is also mechanical pressure base, and these are also this phenomena is also called as mechanotransduction. Mechanotransduction relies on the ability of cell to convert mechanical cues such as stretch or compression to biochemical signals. One way this occurs through the activity of the mechanically gated ion channel. So जो stretch है किसी भी जो प्रेशर डेवलप होता है मैकेनिकल प्रेशर है उसकी वजह से वो ओपन होते हैं और देन mean that is kind of a signal which is received by those channels. So there are also some the channels which are ion specific, potassium channels, the chloride channels we have. There are also anion channel, chloride channel as I mentioned, and nitrate channels as well. So there are specific channels at specific organs, like at stomata, there are potassium inward rectifying channels, potassium outward rectifying channels. ये वो channels हैं potassium inward rectifying channel के जब stomata ने open होना होता है, so they allow potassium to get into the guard cells, जिससे वो open हो जाता है और potassium outward rectifying channel जो है जब स्टोमेटा क्लोज करना है तो वो गार्ड सेल से बाहर निकल जाता है पोटेशियम थ्रू KORC जिसकी वजह से वो स्टोमेटा जो है वो क्लोज हो जाते हैं तो इट मींस जो स्पेसिफिक ऑर्गन्स हैं प्लांट के वहां पे स्पेसिफिक चैनल्स भी हो सकती है वाटर मॉलिक्यूल के लिए सो देयर आर आल्सो स्पेसिफिक चैनल्स aquaporins these are called as aquaporine and these are recently discovered and the Nobel Prize was given those. So it means वाटर के लिए भी जो है वो channels हैं. So then we have carriers. Carriers, as I mentioned, they do not have pores like channels. They need to change the conformation for the transport of ions. यहाँ से that can be passive or active and transport through these carriers is 10 raised to power 2 to 3 molecules. उसमें तकरीबन 1 million or more than 1 million जो है 100 million तक पास हो जाते हैं तो यहाँ पे 100 से 1000 ions per second मूव कर सकते हैं through these carriers. Movement ion through carrier is based on the conformation changes in the transport proteins in both active and passive. दोनों सूरतों में अगर वो passive transport कर रहे हैं या active तो वो उसकी कंफर्मेशन चेंज के थ्रू ही होना this I've already explained that they can be uniport, symport or antiport. ये हम पिछली स्लाइड में we have already explained that what is uniport, what is symport, what is antiport. So then we have ion pumps. Ion pumps they use energy and they transport the ion against the electrochemical gradient. Energetical uphill transport is coupled to another energy releasing event. जब वो develop हो जाएगा charge gradient due to movement of H ion, for example, so then that build up, energy build up will be utilized for the use of another energy taking process that is the movement of ions from lower concentration to higher concentration. So overall free energy change is negative. Ion transport, so this is very important to understand this phenomena that these proton pumps are very important for plants. They pump ions from inside the cell to the outside the cell and they develop a charge gradient and this charge gradient is used for the uptake of the nutrients. So this is very important to understand. There is another factor which is very important and a significant role in plant growth. So that is the pH level. The pH level of apoplast is reduced and due to the movement of H ions from inside to the outside in the apoplast. So you can see here that the pH is low in the apoplast and is high in the symplast or cytoplasm. And this low pH in the apoplast is also important for the extension growth. because the growth is dependent on the flexibility or extensibility of the cell wall. And this low pH in the apoplast, so this that determines the extensibility of the cell wall. So therefore, there are two major significant or important facts in plant growth for this proton pump. That is, the one is that it develops the charge gradient, which you use for the uptake of nutrients. And the second one that it lowers the pH of apoplast for extension growth of cell wall. So these are just animal cell and plant cells. We have a sodium potassium pump in the plant animal cell, but we don't have sodium potassium pump in plant cell. We have proton pumps only here, and these proton pump they develop gradient. मतलब जो काम पोटेशियम सोडियम ने प्लांट में करती है, ये इनके पंप करते हैं वो डायरेक्टली वो करते हैं, तो ये प्लांट के अंदर इनडायरेक्ट चार्ज डेवलप होता है देन विच इज यूज बाय दो ट्रांसपोर्ट प्रोटीन. Primary active transport, yeah, I think we have already explained. The primary active transport is reserved for active transport, which is directly driven by the energy enriched metabolites such as ATP. Uh, ATP used, and this these proton pump they develop, they move ions, H ions from inside to outside, and then this charge gradient develops the outside of the cell membrane, so that is utilized for the movement of other ions like sucrose, other ions as well, and sucrose is also a molecule. Uh, Other ions, as I mentioned in the previous slide, potassium, they can also move along with this H ions from outside to inside. So this is a secondary active transport. So primary active transport, this where a direct energy is used, but here and the energy is not directly used, but the energy utilizing phenomena which develop this